Good afternoon, everyone. My name is Christopher Hattlesey, and I am the Communications Coordinator of Parkinson's Society Ontario. Here with me today is Dr. Naomi Visanji, a neuroscientist that specializes in Parkinson's disease and is researching the relevance of premotor symptoms in PD. I would ask that you hold all your questions for Naomi until the end of the presentation, at which time you can type them in the bottom right and I'll read them out. And now we can begin whenever you're ready, Naomi.
Okay, good afternoon, everybody, and thanks for logging in today. I just want to start by thanking the Parkinson's Society for inviting me to talk to you today. It's always a pleasure to be able to speak to people in the Parkinson's community. And it's my first ever webinar, so hopefully it all goes ahead without any technical glitches. And yeah, as Chris introduced me, I'm a researcher. I'm at the Morton and Gloria Shulman Movement Disorders Centre at Toronto Western Hospital, and I've been in Parkinson's disease research for about 15 years now, and started my life as a basic scientist in the lab, but now I've moved into the clinic where we're doing a lot of work with patients and with clinical trials. And so I'm going to talk to you today about the relevance of premotor symptoms in Parkinson's disease. And let's get started. Okay, so we'll just start with a little bit of basics about Parkinson's disease. Um, it was first described in 1817 by James Parkinson in this essay on the shaking palsy that you can see a picture of here. And beneath that, you can see a quote taken from the essay where he's describing Parkinson's disease for the first time. And a more modern clinical description is that we consider Parkinson's disease to be characterized by a triad of classical motor features, and that's bradykinesia, akinesia, and tremor. And what I want you to take home from this is really that ever since its first description, and even up until modern times, we've typically always considered Parkinson's disease to be a movement disorder. But we're becoming increasingly aware, and I'm sure many of you that have the disease are very aware, that Parkinson's disease also has a lot of non motor symptoms. It really is a very wide ranging disease. And some of these non motor features include things like constipation, sleep problems, problems with vision and smell, some mood disorders, depression, anxiety, and there's autonomic dysfunction as well. And what's particularly interesting about these non motor symptoms is that a lot of them actually can occur very, very early in the disease, and in some cases, long before people actually have any of the motor problems at all. And so, really, all the way through the talk, I want you to keep in mind that the relevance of these premotor symptoms or these non motor symptoms is that they're early. They often occur before the motor symptoms and are called premotor symptoms. And maybe we can use them to try and identify people that are at risk of developing Parkinson's disease. So, I just want to talk very, very briefly about what's actually causing the movement problems. And then we're going to kind of move backwards from there because the two are somewhat linked. And this slide here is just showing you how normal, healthy movement is generated. And it involves a part of the brain, a circuitry called the basal ganglia. And you'll see on the right here, there's this area called the striatum, this large green square. And that area, the striatum, it really is central to everything. It's a junction box that allows everything to be kind of put together and allows movement to occur in a proper way. So it receives inputs from the cortex when you're thinking about wanting to move. And then it also receives input from this area called the substantia nigra. And this is very, very important in Parkinson's disease. So the substantia nigra, it means black substance, and it's called that because the cells there create this black pigment. They're very characteristic. And what these nigral cells do, they project to the striatum and they release a neurotransmitter called dopamine into the striatum. And what the dopamine does is it allows all the cells in this junction box to respond properly to all of the different information that they're receiving, and it allows them to then project properly out to the rest of the basal ganglia so that your movements are nice and smooth and occur the way you want them to occur. So, what happens in Parkinson's disease is that this pathway, this nigra to the striatum pathway, is degenerated. So what you're looking at here on the left is a normal healthy person's substantia nigra. You can see those black strips. That's the pigment that the cells there are producing. And then these areas project down to the striatum. So beneath, what you're seeing is a scan of a person, and those red areas of the brain, that's the dopamine. This person has been injected with a tracer that allows us to visualize the dopamine. So the nigral cells are there and they're projecting to the striatum, they're releasing dopamine, and everything's okay. And just that's popped up there on the left is just an image taken of a nigra looking through a microscope. Hopefully you can see lots of those black cells are present. In Parkinson's disease, you can see immediately that the nigra is not as black, it's not as dark as the healthy person's nigra. And that's because these cells here have literally started to degenerate and be degraded. And what this means for the striatum, when we have a look at the dopamine levels in the striatum, they're much, much lower. And then the striatum is not able to act in the way that it wants to act in controlling movement. And finally, when we look down the microscope, we can see that there's many, many less of these cells around. So a lot of them have disappeared. So what's causing the cells to disappear? A big clue is this thing on the right here. It's called a Lewy body. So you can see this circle with a halo around it. That is a Lewy body, and it's sitting in the middle of a dopamine neuron in the nigra. And they're named after the guy circled on the left, Frederick Lewy, who described these Lewy bodies. In 1919, he published a paper, so nearly 100 years ago now, saying that he saw a lot of these Lewy bodies in the dopamine neurons in the nigra. And it wasn't until nearly 80 years after that, so in 1997, scientists finally managed to figure out what's inside this Lewy body. So what's the Lewy body made up of? And they found out that it's this protein called alpha-synuclein. And alpha-synuclein you're going to hear a lot about in this talk. You've probably heard a lot about it already. But the reason we're so interested in it in Parkinson's disease is because it's basically the main component of these Lewy bodies. And synuclein usually exists on its own. Healthy people have synuclein in their bodies. But in Parkinson's disease, it tends to clump together and it becomes insoluble. And this really has become the kind of pathological hallmark of the disease. In fact, although people receive a diagnosis of Parkinson's disease, we really don't consider that diagnosis to be definite until you take a look and you see the Lewy bodies in the brain. So there's been a lot of research regarding Lewy bodies, and a German neuroanatomist called Heiko Braak, he took the brains of many, many people with Parkinson's disease, all at different stages of the disease, and he started to look at where the Lewy bodies were found in the brain. And he saw that there was a really clear pattern in the spread of the Lewy bodies throughout the brain, and that this correlated somewhat with the symptoms that the people were experiencing. And in stage 1, which is going to be the stage that we're mostly going to be speaking about today, the Lewy bodies aren't found all over the brain. They're not even found in the nigra at this point, but they're found in a region of the brain called the medulla oblongata, which is deep in the brainstem. And then they're also found in an area called the olfactory bulb, which is where we process smell. And at these stages, stage 1 and stage 2, they're called preclinical. And basically what that means is there's no symptoms of Parkinson's disease yet, or certainly not motor symptoms of Parkinson's disease. And perhaps when you ask people in retrospect, did you lose your sense of smell several years ago, then people might realize that they had, but they're not necessarily noticing that anything's going on at that time.
Naomi, sorry to interrupt, but could you put the mic a little closer to your mouth or speak up a little bit? I've had a couple of requests that the volume's a little low.
Yeah, hold on a second. Is that a bit better?
Much louder. Thank you.
Okay. So as the disease progresses, we get to stage 3 and stage 4. These Lewy bodies now are starting to appear in the substantia nigra, so in that region that's so crucial for movement. And this is around the time that people will start to experience those classic motor symptoms of Parkinson's, and end up going to their family doctors. And then finally, as the disease progresses further, the Lewy bodies can progress to other brain regions, places that are involved in emotion and motivation, places that are involved in cognition, and this is when some people can start to experience dementia. So this is a very well accepted theory, and it's really now backed up by a lot of experiments in the lab where they've shown that alpha-synuclein itself is able to actually spread. So alpha-synuclein can spread from one cell to another in a dish, in a culture dish, in an experiment. If we inject Lewy bodies into a mouse's brain, they will start to spread and the mouse will start to develop Lewy pathology throughout its brain. And very importantly, this has also been shown in human brains. So there were a group of patients in a clinical trial who were injected with embryonic dopamine neurons. And so these are neurons from an embryo, they're young or healthy. And they found that after about 15 years, these young healthy cells also started to show Lewy bodies and this synuclein aggregation. So it does seem very likely that alpha-synuclein itself is able to spread throughout the brain. So regardless of these mechanisms of spread, there are a couple of really important and very hopeful things. The first thing is that we know that the Parkinson's symptoms don't manifest until about 50 to 60% of these nigral dopamine neurons are actually lost. And that equates to about a 70 to 80% loss of striatal dopamine. We also know from this then that the brain has a huge capacity to compensate for cell loss. And this is very important. When we talk about a neuroprotective therapy, it might not be that we need to get everybody back to 100%, but maybe we need to just get them to just the other side of this threshold of around 50 to 60%, and then get it to the point where the brain can be able to compensate. And, that makes the target seem a little bit more achievable. The other thing that's very important about this is that by the time your symptoms appear, the disease has been present for several years. So the trick here really is that if we can identify people very, very early, before they've even got these motor symptoms, perhaps we can start to then test treatments at that early stage. And when we've got a good treatment, if we can identify people, we can give them a treatment really early on, and maybe we can stave off them ever even developing some of the motor symptoms of the disease. So there really has been a shift in the field of PD research to find ways of identifying people early. And that's what we're going to now go on to talk about. So we might be able to use some of these non-motor symptoms or premotor symptoms to identify people in the early stages. What we're realizing now is that the pathology of alpha-synuclein being aggregated, it's not restricted just to the substantia nigra or even just to the brain, actually occurs in many different areas. So it can occur in areas of the brain that are involved in mood disorders in very early stages. It also can occur, there have been changes found in the eye, so morphological changes in the fovea. There's been changes in the microvasculature of the eye and in the retina even. And there's many sophisticated machines now being developed to look at trying to very sensitively evaluate the eye in Parkinson's disease and in early Parkinson's disease. This abnormal accumulation of synuclein can also occur in areas of the brain that control sleep. And we're going to talk a lot about a particular sleep disorder. Olfactory dysfunction, we already mentioned that the olfactory bulb is affected very, very early, so smell can be affected. Constipation, Lewy bodies have been described throughout the gastrointestinal tract, and we're going to talk about potentially using constipation to identify people with early Parkinson's disease. And then also autonomic dysfunction. So the heart can be very affected. The autonomic control of the heart is affected, and Lewy bodies have also been found in these nerves that innervate the heart. So this recent study just published this year, called the Onset PD study, has really, really helped us in starting to try to understand just how common some of these premotor symptoms are, and also how wide-ranging they are. What they did in this study was they handed out a questionnaire asking people about whether they've experienced any of 31 non-motor symptoms of Parkinson's disease, and then asking them to date back when did they first experience that symptom. So it's been really, really helpful article in figuring out basically the order in which these various symptoms can present. They handed this questionnaire out to 109 people who had just been diagnosed with Parkinson's and 107 healthy individuals. And what they found was that 17 of the 31 non-motor symptoms were more common in patients than controls. So they then focused on these 17 symptoms and they started to break things down into when did they appear. So this table here, you can see a list of the non-motor symptoms, on the left-hand side. And what you see on the right is the percentage in brackets of people who experience those symptoms. And this is just 2 years prior to diagnosis of Parkinson's disease. And you can see in bold, inattention, anhedonia, and apathy, they were experienced in about 70% of the people 2 years before they got Parkinson's disease diagnosis. They then looked at a sort of longer time span, and they noticed that the pattern was different. So different premotor symptoms were occurring at this stage. And if you look, taste loss, 91% of people were experiencing taste loss 2 to 10 years prior to diagnosis with Parkinson's disease. And then finally on the right here, this is a column of features that were experienced more than 10 years prior to Parkinson's disease. And again, the pattern is a little different. You can see here constipation. Constipation was experienced by 63% of the people more than 10 years prior to diagnosis. Also, you can see that some dream enacting behavior and frequent nightmares, they also occurred in 50-60% of people. So in response to this sort of growing understanding that there really are many, many non-motor features of Parkinson's disease and many that occur long before the motor disorder, a task force was put together by the Movement Disorders Society to discuss whether it's time to redefine Parkinson's disease. So to get away from just that pure motor diagnosis. And I'll just read you this excerpt from this paper. But it basically says, with advances in knowledge, disease boundaries may change. And occasionally these changes are at such a magnitude that they require a redefinition of the disease. And later they say, the recognition of a non-motor prodrome of Parkinson's disease requires that new diagnostic criteria for early stage and prodromal Parkinson's disease should be created. So the field is really starting to shift now and accept that Parkinson's disease is a much bigger disease than just a motor disorder. And we're really starting to recognize these early premotor features. And this task force will now work on defining what those early stage features should be and how can we use them to aid diagnosis. So we're now going to take each of the prodromal features of Parkinson's and discuss the relevance of them. The relevance of them in terms of being able to detect people who are at risk and also use in potential future clinical trials. So we're going to first talk about constipation. And I mentioned that these Lewy bodies, they are found throughout the gastrointestinal tract. I know that most, sorry, I skipped a slide. Okay, I'll just start here. So yeah, constipation, those of you with the disease are probably well aware of how prevalent constipation is, but it occurs in about 80 to 90% of individuals with Parkinson's disease. And that makes it basically one of the most prevalent non-motor symptoms. Several studies have also shown that individuals with constipation who don't yet have Parkinson's disease, they have an increased risk of developing the disease. The paper that I'm highlighting here is reporting findings from a study called the Honolulu Asia Aging Study. And this was a long-term study that was carried out in elderly Japanese American men. And in this part of the study, they collected information on the frequency of bowel movements in nearly 7,000 people who did not have Parkinson's disease. And then they followed them up over a 24-year period, and they noted those that went on to develop Parkinson's disease. So on the left, you can see the number of bowel movements per day. The top row is less than one bowel movement per day, so these people would be considered as having constipation. And then I've put on the right in red the percentage of people who then went on to develop Parkinson's disease over a 24-year period. And you can see that the people with the lowest number of bowel movements, so less than one per day, they had a 3.46% chance of going on to develop Parkinson's disease over 24 years. Whereas those that had one, two, or even greater than two bowel movements per day, that risk starts to drop off. And it's a very, very clear pattern. Now, what's particularly interesting about constipation and Parkinson's is also the time of onset of constipation and then the development of Parkinson's disease. The onset PD study showed us that a large percentage of people had constipation even 10 years prior to Parkinson's disease. And several other studies have looked into this timing, and they're noting a lag of up to 15 years. And that would make this a particularly interesting and helpful feature. If we could detect people 10 or 15 years before Parkinson's disease, then we'd be in really good shape. It is also really important, though, to understand that having constipation doesn't mean you have Parkinson's disease, and it doesn't mean you're definitely going to go and get Parkinson's disease. So if you take a look, even in the people in this study who were constipated and had less than one bowel movement per day, a good 96% of them did not develop Parkinson's disease over the 24-year follow-up. So there's many, many causes of constipation, and because it's so prevalent in the general population, it has what we call a poor positive predictive value for Parkinson's disease. So that's one of the big challenges of using these premotor symptoms, that they're not all able to predict everybody. You're going to get a lot of false negatives. So what could be done to try and improve the predictive value? We know that constipation is involved. We've got enough information for that, but it's got poor positive predictive value. Well, what we could do is try to look for alpha-synuclein in the colon. So the enteric nervous system, which is the part of the nervous system that controls the gastrointestinal tract, is actually often referred to as the second brain. And I know a lot of people wouldn't want to think that their brain is in their bowels, but there's over 100 million neurons that govern the function of your gastrointestinal tract. So it's a lot of neurons outside of the brain. And many studies have been investigating the potential to look at alpha-synuclein in these neurons, basically due to the relative ease of access. And by that, what I mean is that around the age of 50 to 60, people start to have colonoscopies for cancer screening. And at that point, it can be quite simple to obtain some tissue. You're obtaining it from people of the exact age that would be wanting to test people and potentially look for alpha-synuclein. So this slide here is showing you where in the colon alpha-synuclein has been described in Parkinson's disease. So if we take the top part, this darker green part, this is the muscularis, the kind of outer part of the tube, if you consider your colon to be a tube. And in this area, the muscularis, there's this thing called the myenteric plexus, which is full of neurons. So the top row in dark green of the table shows you what has been reported in Parkinson's disease in this myenteric plexus. And you can see all the way back in 1988, in post-mortem tissue, somebody described a Lewy body in a person with Parkinson's disease in this area. And more recently, people have found, even in 10 out of 10 people, they were able to see alpha-synuclein in this area. And this is all post-mortem tissue because this plexus is quite deep, and it's not usually safe to biopsy this deep in a living person. But then when you move into the submucosa, so this is this paler green area, you can look at the submucosal plexus, and this tissue can be obtained during a biopsy. And a lot of people have been looking in submucosal plexus and found that they can see alpha-synuclein in 21 out of 29 people with Parkinson's disease, or 4 out of 9 in another study, 10 out of 10 in another study. And then finally, the most inner part is the mucosa. And you have these tiny little mucosal nerve fibers. And again, people have been reporting they can see alpha-synuclein in this area. And there was one particularly exciting study by Shannon and colleagues where they actually were able to obtain some biopsy tissue that was collected from people a few years before they got Parkinson's disease. So they had colonoscopies completely unrelated a few years before they got Parkinson's. And when they looked in the tissue of these people, lo and behold, they were also able to see the alpha-synuclein. And this got the field very, very excited that we might be able to detect Parkinson's using these biopsies long before people started to actually have the movement disorders. Unfortunately, though, when we look in control people, so these are healthy people without Parkinson's disease, some people also were reporting that they were seeing abnormal alpha-synuclein in the control tissue. And these, the numbers kind of range. Different people using slightly different techniques are seeing slightly different numbers. Some people don't see it at all. Some people see it in half the population, and some people see it in every sample they look at. So it's a big challenge in our field is inconsistency in the techniques that people use that can cause a lot of confusion in the field. So we also carried out our own biopsy study that we recently published, and this here is just showing you some of our staining, and all of this is tissue from healthy people without Parkinson's disease, and we were able to see alpha-synuclein in the colon biopsies that we collected from healthy individuals. So clearly, with so much inconsistency in the field, some more work is required. And what's actually happening at the moment is that a consortium of people are sharing some tissue, sharing methodologies, and trying to figure out whether there is any promise to this or not, because there was certainly enough out there to suggest this could be promising. But everybody needs to put their heads together and figure out the best ways to do it to see whether it's going to work or not. So just to summarize, constipation is the most common non-motor symptom of Parkinson's disease. About 80% of people experience constipation. It could be one of the earliest premotor symptoms, occurring as long as 15 years prior to motor symptom onset. And the Lewy pathology in colonic biopsies may or may not make a useful biomarker for Parkinson's disease. So now let's move on to another non-motor symptom of Parkinson's, and that's sleep disorders. There are several regions of the brain that are involved in sleep that have also been shown to be affected by Lewy pathology and Parkinson's disease. And we're going to talk about REM sleep behavior disorder. So this is rapid eye movement behavior disorder, and it's one of the most common sleep disorders in Parkinson's disease. What happens in REM sleep behavior disorder is that the areas of the brain that are responsible for making sure that when we're dreaming, we don't act out our dreams. So they sort of switch off your muscles, if you like, while you're dreaming. These areas are very heavily affected with the Lewy pathology, and these people then start to physically act out the content of their dreams. So, for example, if the person is dreaming that they're playing soccer, they might kick out as if they're kicking a soccer ball, and in some instances, they can injure their bed partners. Now, there are 4 independent RBD cohorts or groups of people with RBD that have all been shown independently to have a very, very strong association with development of Parkinson's disease or another synucleinopathy. So this could be dementia with Lewy bodies or multiple system atrophy. The first report of RBD preceding Parkinson's disease was published back in 1996. And at this stage, in this cohort, 38% of people who had RBD had then gone on to be diagnosed with Parkinson's disease or dementia with Lewy body. The same group 5 years later then followed up and reported what had happened in their population. And by this point, the numbers had increased from 38% all the way up to 65%. So things were really starting to develop. And most recently, the same group reported their 16-year follow-up. And at this point, 81% of those people who had RBD had then gone on to develop a neurodegenerative disease. And this is a very, very strong positive predictive value. And these groups are saying they can't imagine that this number isn't going to rise even from 81%. Maybe at some point it's even going to reach 100%. So very, very strong positive predictive value. This slide here basically illustrates the same sort of concept, and this is from a separate, completely separate cohort. So you can see at the top here, there were 44 patients in the original cohort, and they were all diagnosed with RBD between 1991 and 2003. 2 years later, in 2005, they reported a follow-up, and at this point, 20 of the 44 individuals had gone on to develop a neurodegenerative disease. So that's nearly half. And of those 20, 9 were diagnosed with Parkinson's disease, 6 had dementia with Lewy bodies, 1 had multiple system atrophy, and 4 had mild cognitive impairment. 22 individuals at this point did have RBD, but didn't have Parkinson's disease. They then went on and followed up with the population again in 2012, and by 2012, 16 of those 22 now also had a neurodegenerative disease. So 7 had Parkinson's, 5 had DRB, 4 had mild cognitive impairment. So in this study, of the original 44, only 4 by the latest follow-up did not yet have a diagnosis of Parkinson's disease or another synucleinopathy. And what was very interesting was when they looked at the brains, they did a scan looking for the dopamine in the striatum, like we saw earlier. All 4 of these people had lost that red signal. They were starting to lose the dopamine signal in their striatum, really suggesting that probably they are going to go on to develop a Parkinsonism. So on the right here, you can see what's called a Kaplan-Meier survival plot. And what this plot shows you is that we start at the top here, 100% of people have no neurological disease. And then as time passes and we move to the right, this number starts to dwindle gradually over time. And so at 15 years follow-up, there's not very many people left who don't have a neurological disease. And this graph illustrates 2 really important features of these people with RBD. The first is that of all of the premotor symptoms of Parkinson's disease, RBD has the greatest positive predictive value for the subsequent development of Parkinson's disease. So currently 81%, and that's only expected to get, larger. The second is that there's this long interval between the development of RBD and subsequent development of Parkinson's disease. In another study, they had 172 people with RBD, and 151 of those people had Parkinsonism. And the mean time from RBD onset to Parkinsonism onset was 14.2 years. And in the longest person, it was 29 years. So this really sort of shows you that this population could be very useful for studying in Parkinson's disease. Now, what's particularly interesting about that article was also that they were able to obtain the brains of 3 of the people who had RBD and then Parkinson's disease. And what you're looking at here are sections of the Nigra stained for alpha-synuclein of these 3 people. And hopefully you can spot that there are Lewy bodies, these round circular inclusions with the halo. All 3 people had Lewy bodies. They also had lost some of the pigmented cells. On the right, you can see some pigment escaping from a cell that was degenerating. And these Lewy bodies and Lewy pathology, again, not just found in the substantia nigra, but also found in these areas of the brain that are involved in controlling REM sleep atonia, so switching off your muscles when you sleep. And if you remember back to the Braak staging, these areas are actually involved really, really early in Braak staging, Braak stage 1 and 2, so very, very early in the disease. So again, very, very relevant to Parkinson's disease. RBD itself is quite rare, but the fact that it's got such a strong positive predictive value and that the pathology is so similar to what we see in Parkinson's disease certainly makes this population very, very interesting and very, very useful for Parkinson's disease clinical trials. And it's impossible to talk about this field of RBD and Parkinson's disease without talking about the work of Ron Postuma, who's a researcher in Montreal. He's an incredibly prolific researcher in this subject. And what he's really been doing lately is gearing up the scientific community to get everybody ready to start to use this population for clinical trials that we hope will be relevant also to Parkinson's disease. So this slide here is just showing you one of Ron's latest papers, and it's the results of a 10-year study. And what he was doing in this study was trying to increase the chances of predicting who would develop Parkinson's disease, and to try and identify those people earlier and earlier. So if you look here at just the solid black line, this is one of those same Kaplan-Meier survival plots. And you can see that over time, as time passes and we travel to the right, the number of people who don't have a neurological disease starts to decrease. But what happens if we then combine one of the other premotor features with those people with RBD? So this middle small dotted line shows you what would happen if we took everyone with RBD and then added on them having, for example, smell loss. When we do that, you can see that the graph starts to steepen, and that basically we're able to increase our ability to predict who's going to develop Parkinson's disease. What happens if you then add on a third premotor feature? So now you have RBD, smell loss, and color vision deficits. And if we do that, we can really, really start to increase our ability to predict which individuals are going to develop Parkinson's disease. And this is really a very, very forward-thinking study. What he's trying to do here is make things achievable for a clinical trial. So you can imagine if we want to have a clinical trial of a potentially neuroprotective drug, we need to be able to predict who's going to develop Parkinson's disease so that we can treat them with the drug and then hopefully prevent them developing Parkinson's disease. So if we've got 100 people and only 5 of them are going to develop Parkinson's disease over the period of our trial, let's say it's 3 years long, you can imagine that we're not going to really be able to take home very much from that. We would have to start recruiting thousands of people into a very long-term study before you'd have enough numbers to be able to determine an effect. But what Ron did in this study was show that by adding on these additional features, we can start to get the numbers down to something that's achievable. So if you look here at the top row, it says baseline. These are the people just with RBD. And over a 3-year period, which is a reasonable period for a clinical trial, 30% of those people would be predicted to develop Parkinson's disease. And this would mean if we had a moderately effective neuroprotective drug, Ron estimates you'd need about 134 people with RBD in your study to see a bit of an effect. And then if you look at this bottom line here, we take it to all 3 things being abnormal, so RBD, olfaction, and color vision. Now 64% of those people with those 3 premotor features are going to go on to develop Parkinson's disease. So we can pretty much halve the number of people that are needed in order to see an effect. So this is a really great concept that's been receiving a lot of attention, and he's really, really readying the field to when we've got a neuroprotective therapy to trial, this population might be one of the ideal populations to study it in. So just to summarize RBD, it's the strongest premotor symptom predicting Parkinson's. There's Lewy pathology in the regions of the brain that control REM sleep atonia. And this population could be very, very valuable for future clinical trials looking at neuroprotective therapies. So I'm now going to move on to olfactory dysfunction quickly. And this has been described in up to 80% of individuals with Parkinson's disease. It's important to note that not everybody with olfactory dysfunction is aware of it. Sometimes people don't realize until long after they've lost their sense of smell. But the incidence is so great that I've even seen some papers where people have suggested if you think someone's got Parkinson's disease but their smell is intact, you might have to reconsider the diagnosis. It's very, very strongly associated with Parkinson's. And we've discussed that the Braque staging shows that the olfactory bulb is affected very, very early. There's several commercial tests, so olfaction can be tested very, very easily. That's a good thing for a potential biomarker. And some of you that have been in clinical trials might even recognize these things. There's the UPSIT test, scratch and sniff booklet on the left, and on the right there's the sniffing sticks where you wear a blindfold and you smell things and write down what you smelled. And prospective studies have shown that there's a correlation between olfactory dysfunction and later development of Parkinson's disease. So again, that Honolulu Asia aging study that we talked about with constipation, they showed that they had 19 people develop Parkinson's disease over a 4-year period. And when they looked back over 4 years before, what was those people's sense of smell like, they found that they all had a deficit on these tests. So there's pretty compelling evidence that hyposmia is a prevalent non-motor feature of Parkinson's disease and that it can occur before the disease, the motor symptoms manifest. But there are also lots of caveats to using a simple smell test to predict who might develop Parkinson's disease. The first thing is that, surprising, even though we've got these Lewy bodies in the olfactory bulb really early in Braak stage 1, the time that we actually get olfactory dysfunction prior to Parkinson's disease is estimated at only being about 4 years. So again, in the Honolulu Asia aging study, if they looked at people's smell scores more than 4 years before they were diagnosed with Parkinson's disease, they didn't see any deficit. That deficit didn't start to happen until closer to their diagnosis with Parkinson's. And this is probably because you can have Lewy bodies and alpha-synuclein aggregating, but it doesn't necessarily mean that the cells are dying and that the cells there are dysfunctional at that point. That might happen at a slower rate. The second is, similar to constipation, olfactory dysfunction has a very high frequency in the general population. So again, we have that very, very poor positive predictive value. And again, much like constipation, you could try to improve this positive predictive value by also biopsying this area. And this study here shows that there was Lewy pathology. This is in postmortem tissue, but they saw Lewy pathology in the olfactory bulbs. You can see the Lewy bodies here on the left in panel D. These are Lewy neurites in E, and in F you can see a low power image just showing you the extent of this synuclein deposition. It's this sort of bluey colored deposition. And this group showed that they found Lewy bodies in 55 out of 58 people with Parkinson's and 34 out of 35 people with dementia with Lewy bodies, but only in 5 out of 69 controls. So that's quite a good separation there. The one problem is, though, olfactory bulb biopsy is a very invasive procedure in itself. So these authors weren't suggesting that everybody gets biopsied, at age 55 or something to look for Lewy bodies. But what they did suggest is perhaps you could use this kind of technique to confirm a diagnosis in somebody that might be considering a very invasive therapy, for example, DBS surgery. So if you were unsure about the diagnosis and you weren't sure that DBS was the right option, perhaps these people, the risk of the biopsy itself would be worth it to confirm a diagnosis. So in summary, olfactory dysfunction is very common in Parkinson's disease. The olfactory bulb is involved very, very early, but surprisingly, the premotor interval of the actual symptoms of smell loss is quite short, so might be superseded by something else that happens earlier on in the disease. Now we're going to talk about cardiac autonomic dysfunction, and Lewy pathology has been found in the cardiac nerves in Parkinson's disease. And in this study, Arimo and colleagues, they managed to obtain cardiac tissue from people, controls, people with something that's called incidental Lewy body disease. Now, incidental Lewy body disease is considered by many just to be a precursor to Parkinson's disease. Basically, you've got Lewy bodies but no symptoms. And they also collected tissue from people with Parkinson's disease and multiple system atrophy. And what you're looking at here are some cell bodies that are in an area called the paravertebral ganglia. And these cell bodies, they send these long axons all the way to the myocardium, or the heart tissue, the heart muscle. And Arimo and colleagues then took these long axons and the ends of them and the cell bodies, and they looked for alpha-synuclein. And in the control population, they found no evidence at all of any synuclein aggregation. In the people with Lewy body disease, they found that there was alpha-synuclein in the axons, but not so much in the cell bodies. In Parkinson's disease, they found that the axons are starting to degenerate, and at this point, the synuclein is lost from this area and is found predominantly in the cell body. And then finally, in MSA, they found no evidence of synuclein aggregation. And what they took away from this was that there's this sort of pattern of where the synuclein goes, and then it leaves there and is followed by a neurodegeneration. And they called this a distal dominant degeneration. So these distal far areas of the nerves by the myocardium were actually having the synuclein deposition first and then degenerating. And then the synuclein deposition is moving on to the cell bodies. So a lot of people are very interested and think that this might mean that detecting synuclein or looking at these distal axons in the heart might be useful in very early Parkinson's disease. And we're currently carrying out a study to try and investigate this at Toronto Western. And we're using a radioligand, so MIBG. It's radio labeled, and the MIBG is injected, and then it's taken up into the cardiac nerves. And using a SPECT scanner, we're able to then scan the heart and have a look at what these nerves look like in the heart. Our study is going to include 50 participants. We'll have 30 people with Parkinson's disease, all at different times, some of them very early, 10 with atypical Parkinsonism, so MSA and PSP, and 10 healthy individuals. And they'll be injected with the MIBG and then imaged at 3 time points afterwards. So what I can show you here is some preliminary data from our study. So we've scanned to date 9 of the 50 people that we're hoping to scan. And this is an image from a healthy control. And what you're seeing is all of the images of the heart that the SPECT scanner takes. So any of you that have had a SPECT scan, you'll know that it's got multiple cameras and they kind of rotate slowly around your body, taking images, so you get this 3-dimensional structure. And what we do is we take each of those images and then we compress them down to form what's called a polar plot, which is this image on the right. And so this basically is a representation of all of the nerves that have taken up our scanning substance in the heart, all compressed onto each other. And you can see here that there's a lovely uptake throughout the whole heart at the early time point, and even 24 hours later, we've got really, really good uptake. So this suggests that the person's nerves are in really good shape. This is the images of a person with Parkinson's disease who was in our study. And hopefully you can see that even immediately, at the immediate time point, there are areas of this person's heart that really don't have very good signal, so that the nerves here are damaged in some way and they haven't taken up our scanning substance. And it's very, very obvious here at the later time point that this has happened. So we're now hoping to extend this study. We'll carry it out in more people, but we're very, very excited by our preliminary findings. So just to summarize the talk pretty much, we've talked about 4 of these premotor symptoms of Parkinson's disease and how they might be useful in identifying people early. We talked about REM sleep behavior disorder and how useful that population might be for clinical trials. We've talked about constipation and how prevalent it is, but also that it has poor positive predictive value, and maybe we can try to use biopsies to try and enhance that. Similarly, olfactory dysfunction, poor positive predictive value, also maybe a very short prodromal interval, might only be 4 years, which might not be early enough. And the cardiovascular autonomic dysfunction, where really we're hoping that some of the advances in technology and scanning techniques will allow us to maybe try to detect that very, very early on, because certainly the pathological studies would suggest that these axons in the heart might be affected very early. So again, I just want to leave you with this point. This is really what is the relevance of these things. And it's just to talk about the neuroprotective clinical trials that everybody is so hopeful for. And what everybody needs to try to understand really is that a neuroprotective therapy, to be successful, it probably needs something to work with. If you remember those pictures of the Nigra, when we look at the Nigra here of a healthy person, there's lots and lots of cells there to work with. But by the time somebody has got sort of end-stage Parkinson's disease, we've lost a lot of those cells. So any neuroprotective therapy, it just might not have anything to work with. So we really need to be getting people early on in the disease so that we can give these therapies their best chance of working. So to date, neuroprotective trials in many diseases have failed, and it's probably basically because they're not being given the chance to work, they're not being given early enough. So that's why there's this huge push now, really, to try and identify people early. So I don't want to skip forward to this slide here that shows you what are the latest trials that are happening. And so I went on clinicaltrials.gov last week, and I found that there are 495 studies actively recruiting for Parkinson's disease, which is great news. And then I noticed that 2 of the top 3 are all about predicting who's going to develop Parkinson's disease. So this first one here, development of clinical prognostic models of Parkinson's disease. So how can we predict who's going to get Parkinson's? Number 3 here, biomarker discovery in Parkinson's disease. So this really is a very, very active field of research, and with very good reason. So I just want to end by thanking everybody for listening and acknowledging my colleagues who, I have a picture here of my colleagues that aren't showing up. So anyway, maybe you can see them, maybe you can't. But I just want to acknowledge my wonderful colleagues at the Movement Disorders Centre, particularly Dr. Connie Maris. Most of the content of what we've covered here, Connie and I have just written a review about. So she's had a huge amount of input in the content of these slides. So yeah, just thank everybody for their time. And if anyone's got any questions, feel free to ask.
So the first question is, no loss of smell but having Parkinson's disease, is it possible?
It is possible. Yeah, it definitely is possible. I think, a lot of people do lose their sense of smell, around 80%. I've been surprised when I've read some people suggesting that if somebody has an intact sense of smell, the diagnosis should be reconsidered. And an anecdotal thing, I have absolutely no idea if this is possible or not, but my mother, who has Parkinson's disease, she's not had a sense of smell for many, many years. But the other day texted me to tell me that she could smell a particularly strong lily in the back garden, so that she had never been able to smell in recent times. So I think sense of smell is, it's somewhat subjective, and sometimes it can be subtle. The olfactory dysfunction we talk about in Parkinson's disease, sometimes the patient may not be aware of it, but when we test them with the sniffing sticks or the upset, a very sensitive test, you can start to weed things out that maybe the person's not aware of. But yeah, I would say it's not impossible.
We have another question. Any benefit from low intensity laser therapy?
I don't know anything about that, I'm afraid. Yeah, I would need more information, but I haven't heard of that therapy, so it's not something that that's particularly well known, I wouldn't have thought.
What, of any, studies that surround prevention of Parkinson's disease, i.e. lifestyle, nutrition, etc.?
Yeah, I mean, there's all kinds of studies looking at environmental factors, including diet, smoking, caffeine, um, exposure to various things in your life, like pesticides and stuff like that. There are definitely a lot of environmental factors that contribute to risk for Parkinson's disease. So, I mean, that's a whole nother talk in itself. But undoubtedly, it's not just genetics at play. There are multiple causes of Parkinson's disease, and many, many factors at play. So, there's too many to list. But I mean, caffeine is one that's actually being studied at the moment in a clinical trial at our centre. It's a multinational clinical trial, but they're enrolling people who will, rather than just drinking coffee, these people are going to take a caffeine pill, every day for I think 5 years to see if it does anything to the progression of the disease. So caffeine is up there.
As for the studies that you mentioned at the end, how does one register for those studies?
So there's multiple sources of information. So clinicaltrials.gov will give you all of the registered trials. A lot of those are in the States. You can also find out about trials from places like the Michael J. Fox Foundation has a good section on clinical trials that are registering people. They have a sign-up thing called Fox Trial Finder, where you can actually log on and put in all your information, and then they'll match you with studies that are recruiting. So it's sort of like a dating site for people who want to participate in clinical trials, and then people like us that want to recruit them. Anyone who's local to the Toronto area, you can contact our clinic absolutely to ask us about trials that are recruiting. And a lot of people, your neurologists they might surprise you by how much research they're actually involved in. So you can always speak to your neurologist and express an interest to be in a trial. And I bet you they'd either have one that you might be interested in participating in, or be able to pass on your information to somebody that was recruiting. So it's we're always when people are interested in participating in the trials, and hugely grateful for the people that do.
Okay, so we've run out of time. That concludes the webinar. I want to thank you and everyone for attending, and special thanks to Naomi for taking the time to do this. Very much appreciated. I will be uploading the webinar to our website, hopefully by the end of today, but if not today, tomorrow. And if you have any questions at all, everyone should have my email. I will type it out into the chat box. So thank you to everyone, and enjoy the rest of your day.
Thank you. Bye-bye.